to have special guests here for our March roundtable. And as is our custom, we'll let our speakers give their presentation, but you should feel free to chime in, and that goes equally to the researchers and to the families. That's sort of our quasi-lab meeting style here. So don't feel like you have to save all your questions for the end. If you're confused by a point, you're probably not alone. So please feel free to avail yourself of the chat if that's your preference, or just wait for a pause and just jump in. So with that, really excited to have two speakers, two guests today, both working in the project under the Project AP researcher umbrella. So Dr. Francesca Telese, Dr. Yanning Zuo, can you please introduce yourselves? Maybe Francesca, you go first. And yeah, we're very honored and excited to have you here today.
Yeah, it's my pleasure to be here. Thank you for the invitation. And my name is Francesca Telese. I'm an associate professor at the Department of Psychiatry at the School of Medicine at UC San Diego. And my lab is interested in gene regulation in the brain, and we focus on several neuropsychiatric disorders, mostly substance use disorders, but also associated traits, which fall under the umbrella of also neurodevelopmental disorders. And Yanning is a postdoc in my lab. She's an expert in computational analysis of single cell gene expression sequencing data, and she has a very strong interest in neurodevelopmental disorders, and this is one of the reasons she's working on this project. And another reason was that my lab has been working on this protein called reelin. Reelin is a glycoprotein that's produced during brain development at different stages. It's very important during corticogenesis as it guides the migration of newborn neurons, but it also has an important function in postnatal development, although less studied or understood, especially in the very first postnatal stages. And since we had heard about preliminary data from your Substack a few months ago, maybe at the beginning of the year, and of the exciting preliminary data that reelin was one of the really positive cells had been identified as altered or overexpressed in organoids from 8P families. That's when we started this discussion, since we have been studied the role of reelin in brain development, and that's how we got all connected. And we are really honored and happy to contribute to this project, and hopefully to we actually now our Yanning results have branched out beyond reelin, and she will show you her results today. So Yanning, go ahead.
Yeah, thank you, Francesca, for your introduction. So let me first share my screen. There we go. Can we hear the screen? Can we see the screen?
Yes.
Cool. Awesome. So then I'll get a highlighter for myself. Yeah. So today I'm going to introduce to you my progress on analyzing the single nuclei RNA-seq profiling of the chromosome 8P disorder brain organoid model. So a little bit of kind of to remind you about the background. So I think earlier last year, Dr. Moitra's lab developed a brain organoid model for chromosome 8P disorder derived from iPSC cells from a family tree. So the family tree includes a proband, a female proband, and her parents. And the iPSCs were induced into neuronal fate, and then it was kind of cultured to to develop brain organoids. And in the media, the brain organoids were cultured for a total of 9 months. And there are 4 sample times during the 9 months, which are at 1, 3, 6, and 9 months, to look at how the brain organoids are going at different time points. And then these samples were sent for a single nuclei RNA-seq to look into their transcriptional status. So with the data set in hand, I have mainly 2 aims to probe what's basically going on in the 8P organoids, different cell types. So the first aim is to characterize differentially expressed genes and gene co-expression networks, as well as pathways differentially expressed or showing up in 8P phenotype. And my aim 2 is building up on my aim 1, which is to identify shared molecular mechanisms between 8P disorders and other neurodevelopmental disorders. And if you have any questions, just let me know, at any time of my presentation. So I'm first going to jump into aim 1, but before I'm introducing to you my results, I would like to firstly talk a little bit about the approaches that I'm using. So the 2 main things I'm going to do in this aim 1 is to firstly characterize differentially expressed genes. So usually we call them DEGs, and then construct gene co-expression networks. And the pathway analyses can be applied to both of these 2 kind of gene groups or gene programs to kind of get an idea about what their functions are. So firstly, about the differentially expressed genes, it's kind of like on the individual gene level, and you can use, for example, I'm using the Wilcoxon rank sum test provided by Seurat, which is the really popular package to analyze single cell or single nuclei RNA-seq. So you can use some approach to identify individual genes which are significantly up or down regulated between different conditions. So for example, in our case, the conditions are 2 genotypes. One is control, or the parents control genotype without 8P genotype, and the other is 8P disorder genotype. And another complementary approach to look at transcriptional changes in the data set is to firstly construct gene co-expression modules and see what are the modules significantly correlated with the 8P genotype. So this is using a package called WGC-NA. The whole name is Weighted Gene Co-expression Network Analysis. So this is considered as a really nice complementary because it's not relying on anything from this DEG step, and it rather starts from scratch. Like after you clean your data, you can just input the gene expression matrix into the WGC-NA pipeline. And it constructs biologically relevant networks, even in the absence of significant DEGs, because it rather captures correlated groups of genes. So as long as these genes are kind of co-varying with each other, they would be captured into different groups. And we really call these gene co-expression groups as modules, which means just a group or a module of genes. And this approach was firstly developed for bulk tissue RNA-seq, but it has been adapted to use on to single cell or single nuclei RNA-seq data. So this is I'm basically using a similar approach as one of my previously co-authored paper. I'm going to show you this cell type characterization, cell composition analysis result. So after clustering these cell types, I find 30 distinct cell subtypes of the brain organoids. And here is a UMap of all the cell types. And here I'm also listing out each individual color and their corresponding subtypes and major cell classes. So I use the abbreviations in the cell subtypes, and here the major classes are kind of introduced into what these abbreviations means. So the first major cell class is called cycling neuronal progenitor cells, which the abbreviations are NPCs. So these are considered as a very early stage of neuronal differentiation, and these are rather neuronal stem cells, which can turn into neurons or glial cells. And then the next step down the developmental trajectory are these radioglial cells, and these are RG. So these are kind of already starting to differentiate from the NPCs, and they are still at a rather young stage, and you sometimes can't really even determine whether they are on neuronal fate or glial fate. And then neuroblasts are rather committed cells or very young immature neurons, which will later develop into mature neurons. And then you have excitatory and inhibitory interneurons. And another fate, radioglial cells can go on are called these glioblast cells. It's similar to neuroblast, but these glioblast cells will turn into glial cells in the brain. And also there's another cell type, which are also a non-neuronal cell type called choroid plexus cells. So these cells are making of a membrane like wiping around the brain, and also provides a lot of like physiological supports to the brain. So here, the maps are just showing these more than 75,000 of cells. But actually, when I was firstly trying to annotate the cell types, I got pretty confused because I was told it was a cortical organoid, but actually when I was analyzing the data, I find that more than half of the cells were expressing cerebellum lineage marker, UNC5C, while only a smaller portion of cells are expressing this telecephalon lineage marker, FOXG1. So this is pretty interesting, and I actually looked into many human and other animals, cerebellum, single nuclear RNA-seq to make sure I'm really looking at the right thing. And also I looked at previous cerebellum brain organoid papers to make sure I'm giving these different cells a clear subtype annotation. And here in the lower right corner, I'm showing you how many nuclei counts per cell type. So basically, it's ranking from the very abundant cell, which is interneuron number 1 population of the forebrain, all the way to like the least abundant cell, which is glioblast number 7. And basically, for each cell type, we all have a rather decent number to do later analyses. So any questions at this page? No? Okay, I'm going to show you my cell compositional analysis result. So here, firstly, I'd like to look into these major cell classes at different time points. So if you look at the distribution of these major cell classes across the subjects, you can see that at 1 and 3 months, it looks rather similar. But starting from 6th month, you can see there is a significant change in the GE3, which is the proband. And also at 9 months, it rather looks different between all the individuals. So I performed a statistical analysis using Propeller, and I find that at 6 months, there is a significant increased glioblast proportion in proband. So you have all these as glioblasts, and you have a significant decreased proportions of NPCs, radioglial cells, neuroblast and interneurons in the proband. So basically, you can see her parents have all these cell types, while the proband is only having kind of really minimum of these 4 different cell types. And at 9 months, although the 3 subjects all looks pretty different from each other, but I did successfully capture a significant increase in excitatory neuron proportion and choroid cell proportion in the proband. So, and also I'd like to mention the caveat again, because that's rather n equals to 1. And here I can only kind of put the 2 parents together as controls, and I can't really model a ANOVA analysis with sex as a variable. So after looking into these major cell classes, composition.
Can I ask about this?
Yeah, sure.
So regarding the concern that you have an N of 1 experiment here, if you were just to derive 3 independent IPS lines from the same person and run this experiment, what sort of variability would you see from organoids independently derived from the same person? And like, do these differences you're seeing strike you as, you know, outside what you'd expect from just assay to assay or clone to clone variability?
Yeah, this is a really good question. I think firstly, if you were to develop a iPSC into different organoids, so I would say if it's a dorsal cortical organoid, the kind of protocols are pretty well set, and the studies have demonstrated that you can basically do the same protocol on different like clonal origins and then produce similar proportions of similar cell types. So basically you have a list of cell types pretty nicely defined, and you can see the proportions are similar across different lines. But that case is for dorsal cortical organoids, which are kind of a pretty standardized pipeline. But in this scenario, it's a brain organoid, which is pretty different. And from the papers, I know that brain organoids can vary from each other a lot. So this is also a huge caveat, and we really want to kind of see whether, if possible, I'd like to see more kind of data from different subjects or from different lines to make sure this is replicable. So this is cell compositional changes on the major cell classes. And then I dived into the cell subtype compositional changes. And this is a kind of even messier situation comparing with the major cell types, because each individual cell composition are pretty much different even at the same time point. But you can still see the major cell types are over there, but the cell species are basically just different. You can't even call them as the same cell subtype. So even in this scenario, the propeller package successfully captured some statistical changes at 9 months in the proband, which are significantly increased granule cells, 2 granule cell subtypes, and the choroid cell subtype here, and a radioglial cell subtype, and 1 excitatory neuron subtype. These are kind of squished into these little pieces. So for these two smaller ones, I can't really say for sure whether it really means something, but at least you can see for the choroid number 1 population and the 2 granule cell population, it's a really huge increase comparing with the parents, because the parents don't really have, for example, like the granule cell types at this 9-month time point. So to be specific, these 2 granule cell types are the glutamatergic and really positive cell types we observed in the previous data. And I also kind of matched kind of a bunch of different markers, and I determined that these are granule cells of the cerebellum rather than other cell types because of their distinct expression pattern. And if you're interested, I can kind of dive into another slide, but I didn't really put it in the mainstream of this presentation. So still, because there's basically no biological replication, we can't really say that these data are replicable, but at least I can report that there are significant changes in these 5 cell subtypes at 9 months of age in the proband's brain organoids. Cool. So a little bit of conclusion for the first part. I got more than 75,000 high quality nuclei, and I annotated 30 cell subtypes from this data set. And the cell annotation shows that NPC and RG cells and also the neuronal cell types express markers for both telencephalon and cerebellum, as well as a lot of kind of midbrain markers. But because these are not so defined, I'm basically putting out these 2 major kind of origin, potential of origin or similar to these 2 different lineages. And also I developed, I find significant changes in cell composition between the proband and parents at 6 and 9 months. And also among the cell subtypes with significant compositional change, the really positive cells express markers for granule cells of the cerebellum. And then I'm going to jump to the next part, which is the DEG analysis. So for DEG analysis, I really kind of was challenged in a way, because you can see from this plot that cell subtypes are kind of showing a really high level of subject and temporal heterogeneity. So basically at each individual time point, you have really different cell types, and across the 3 subjects at each time point, you have really kind of different kind of patterns of kind of distribution, because one subject may have one cell subtype, while the other 2 subjects don't really have that cell subtype. So to perform this analysis, I'd rather choose the cell subtypes with at least 100 cells from each subject at the same time point to make sure I'm doing a fair comparison across the 2 conditions and the 3 subjects. So here I still put the 2 parents together as control, and I compare the controls with the proband's genotype. And I use the standard threshold of FDR lower than 5% and log fold change of above 0.25. And this gave me kind of differential gene expressed here, like you can see up and down regulated in these in these 10 time points plus cell types. And if you look at this bar plot, probably you can't really say what are the functional things, and you can only say probably one cell type is showing more differentially expressed genes than others. And then I perform the pathway annotation to look at what are the functions of the DEGs in each cell types at each individual at each individual time point. So for at 1 month, we observed a I would say more of a down-regulated trend of neurodevelopment related terms. While if you look at some cellular signaling pathways and also translation and also, for example, like protein processing, you have basically increased significant DGs in these pathways across these 4 cell types, which includes NPC, 2 RGs, and 1 excitatory neuron. And also you have, I observed some cell communication changes in this pathway as well. And moving from 1 month to 3 months, I think the trend becomes even more kind of obvious, because you can see that basically almost all of the neurodevelopmental terms are down-regulated at 3 months. in these RG, neuroblast, excitatory neuron, Purkinje cell, and the glioblast. And the neuronal terms are showing also made like more of downregulated than upregulated. And also you have downregulated cellular signaling, you have majorly downregulated cell-cell communication. But interestingly, I observed an increase across 4 cell types of the 5 of cholesterol biosynthesis and metabolism of steroids in these cell types, which is rather interesting. And I'm not so sure like why these are showing up here. And moving on to 6 months and 9 months, we basically don't really have a lot of cell types to discuss because at 6 and 9 months, the cells are really different across subjects. And I only have a excitatory number 1 for 6 months and a glioblast number 4 for 9 months, which basically shows a lot of upregulation in neuronal functional terms and cell-cell communication. And at 9 months, we only have a decreased cell-cell adhesion here. And in this scenario, I think I can't really draw a conclusion for the scenario of 6 and 9 months. And I can only safely say that at least at 3 months of the time point, there is a downregulation in neurodevelopment in the proband comparing with the controls. So for a conclusion of this part, firstly, the cell subtypes are showing a high level of subject and temporal heterogeneity, and DEG is characterized with selected cell types, shows a downregulation of neurodevelopment at 3 months of age in the probands. So any question for this part?
Yanning, so how many, was there a difference in the number of interneurons that you had? I'm assuming that was at 9 months?
At 9 months, it's basically pretty much different. So we have a lot of interneuron, but it's only in the controls, and the proband doesn't really have much of the interneurons. It's less than 100, so I can't really do a comparison. And even in control, you can see the JE1 is, I think, mother, JE2 is father. The interneurons are majorly coming from the father, and the mother is only having this little amount of interneuron at 9 months. So
and then the GC cells are, what were the GC cells? Sorry.
Oh, the GC cells are granule cells. So these are excited neurons, but I'd rather use their name of their resemblance to remind myself. So these can be considered as excitatory neurons. Oh,
so
sorry. These are the right. This is right.
Those are
right.
Yeah. So
I'm not sure they got into the DG analysis because they're mostly from the proband. So if there's not enough cells in all the subjects, they cannot be compared. So DEG can only give, partial, insightful information, given the big heterogeneity of cell types among subjects. They can be compared. So I think, as Yannin mentioned, the whole take-home message here from the cell types we can compare, it seems that at 3 months, there's a trend of a reduced expression of genes related to neurogenesis and neural differentiation. That's like the main trend we can gather from this analysis. But probably the next analysis will be more insightful.
Yeah.
Some of it.
Cool. So next analysis is this WGCNA co-expression network analysis. And this is not really based on any DEGs, and it's rather independent analysis. But I do find the results to be very interesting, and it's showing a converging kind of trend with the DEG results. So firstly, after I constructed these results, I constructed these co-expression networks. I performed a gene trait correlation analysis between the ATP genotype and all the modules I have from each individual cell subtypes. And here I kind of have a correlation enrichment score, which you can consider as whether one cell type is having a higher percentage of significantly correlated modules compared with all the significant modules across across the total modules. So basically, we can see that although the significant modules are rather like showing up in almost in each individual cell types, but overlay you have a higher level of enrichment in these excitatory neurons, inhibitory neurons, and glioblast cells, comparing with the MPC, RG, and MB cells. And here on the right side, because I actually there are more than 350 significantly correlated co-expression modules. I basically put them up on this volcano plot to showcase the kind of most significant ones with larger kind of R square, which is correlation coefficient. So here, starting from here, I'd like to firstly dive into some modules, because while I was looking at their annotation, I saw something interesting. So for example, the top 4 down-regulated modules are these glioblast number 2 turquoise, GC granule cell 2 brown 4, GB, which is glioblast number 1 green, and interneuron number 3 red. So these 4 modules. And among the 4 modules, the GC2 brown 4 is not annotatable because there's a lot of non-coding RNAs inside this relatively small module. But across these other 3 top negatively correlated modules, I find that they rather resemble the DGs of glioblast number 2 at 3 months of age. And I actually did a intersection between the GB2 turquoise module and the GB2 DGs at 3 months of culture, and there's actually a lot of shared, or more than half of genes shared, while they still have a 1/3 of genes to be specific to each of these 2 kind of gene programs. And basically, if I saw this many genes overlapping, I would think there must be a lot of functional things overlapping as well. So I performed the pathway annotation to these 3 modules plus I also put the DG at 3 months here as a comparison. So you can see that in the 3 top negatively correlated modules, we have basically everything down-regulated. We can consider is similar to down-regulation of neurodevelopmental terms, neuronal functioning terms, chromatin remodeling, and also the cell communication terms. So this is rather converging with the DEG analyses, while they are 2 totally independent analyses. So this is giving me more confidence to the DG analysis result. So also moving from this part, I'm going to first give you a small conclusion and then show you something more exciting in my aim2. So a conclusion of this part is, firstly, I identified 367 modules significantly correlated with the 8P genotype. And also the co-expression modules kind of result is converging with what we have from the DEG functional annotations. And also the top down-regulated or negatively correlated DEG or modules are enriched in neurodevelopmental and cell-cell communication pathways, and they are in the down-regulation or negative correlation trend in the 8P genotype. The next part is this aim2, which is to identify shared molecular mechanisms between the 8P disorders and other neurodevelopmental disorders. So here I'm using an integrative genomic pipeline called mergeomics to integrate human genetic studies we have from GWAS, or whole exome or whole genome sequencing, to kind of see whether they are enriched in our WGCNA modules or DEG sets. So here, these are the human genetic studies I used. So firstly, I grabbed common variants of 3 phenotypes from 3 different human GWAS studies, which are autism, intelligence, and epilepsy. And while I was reading the papers, I find that for severe developmental disorders, including severe autism or intellectual disability, and also epilepsy, the major factor are in the rare variants. So I also grabbed a few papers from the rare variant side, including ASD, neurodevelopmental disorders putting together, including intellectual disability, and also developmental disorder, including also other issues with the other part of the body. Also, I also find the epilepsy rare variant data set. So to perform this analysis, if I were starting from the GWAS side, I would firstly kind of convert the GWAS loci signals into genes, which is this marker dependence filtering part. So in this part, I'm not only using the GWAS analysis and genes based on and genes related to a locus based on its distance, but I'm also integrating expression and splicing QTLs from the GTAX number 8 version. So basically in this study, I used the expression and splicing QTLs from 4 different brain tissue sets, which I think is really corresponding to the cell types I discovered. And after the MDF step, the GWAS loci would be kind of find correspondence to different genes based on distance mapping and QTL mapping. And from the rare variant side, the things are even more easier. So I just have to put the p-value and the genes as rare variants into this step, and then I can perform MSEA, which marker set enrichment analysis, to see whether some of my modules are enriched with these human genetic signals from either the common variant side or from the rare variant side. So with this in mind, I'm going to show you this part of my result, which is pretty kind of, I'm pretty excited to see this, because I saw the 8P disorders significantly correlated modules are showing a lot of enrichment in terms of different neurodevelopmental disorders. And also the DEGs, to my surprise, is showing kind of even more comparing with the modules. So here, I'm only showing you top 17 modules based on its significant association. Firstly, they are significantly correlated with 8P disorder, and also they are significantly associated at the same time with ASD rare variant, neurodevelopmental disorder rare variant, and developmental disorder rare variant at the same time to be included for this visualization here. Because for other categories, I have rather 30 or 40 different modules, which is rather challenging to perform a visualization. And also I put a DEG sets enrichment here as well. So because we only have 10 DEG sets, so everything's here. So basically, starting from the significant module side, I'm using astrocytes to indicate its strengths of correlation with 8P disorder, and also the direction of correlation, whether it's positive correlation or negative correlation. So going through the annotation of these 17 modules, I find that the net top annotation for the positively correlated modules are rather axon guidance, cell-cell communication, metabolism. And the top annotations for negatively correlated modules are synaptic transmission, cell-cell communication, axon guidance, and also there's a cilium movement, which is pretty interesting to me, and also chromatin remodeling. So these results are rather kind of also agreeing with what we saw in the DEG annotation. And also for DEG set, to my surprise, these DEGs are super enriched for both CVs, common variants, and rare variants of the different developmental disorders, as well as the intelligence trait. So here you can see that for modules, only a few modules are showing kind of high level of or significant common variant enrichment, while the DG set is mostly showing enrichment for both ASD-CV for both common variants and rare variants. So this is pretty interesting to me, and it's suggesting that the modules are a finer dissection of functional processes in cells. So for some modules, these are rather focusing on the core part, while for some modules, it's rather focusing on the peripheral part, which are probably more explained by these common variants. And another thing interestingly is that, remember, for the previous slide, I showed you a high level of overlap between the GB2 turquoise module and the 3-month DEG for GB2. While the 3-month DEG for GB2 is showing nothing for enrichment, the GB2 turquoise module is showing among the highest level of significance across these top modules, significantly correlated with the rare variant. So it's pretty interesting to see how much of signals are kind of missed in the DEG analysis, because there's a hard threshold to kick away the genes below the threshold, while for WGCNA, it's rather putting genes with the same trend of variation in terms of expression into the same group. So this is pretty interesting to me. And the next one is also something I feel pretty excited about, which is I find there are 2 key driver genes shared between the top 8P correlated modules and the DEG, the 10 DEG sets. So the 2 genes are firstly CA-CMB4, which is calcium channel, voltage gated calcium channel, I think. The other is PCLO. So this is a gene functioning in extracellular matrix or something, or I think cell skeleton. Yeah, I think cell skeleton. So these 2 key drivers I find in both DEGs and also these different co-expression modules, these top correlated co-expression modules. So here on the left side, I'm visualizing the Bayesian network of these 2 individual key driver genes. And the Bayesian network is basically the network I used to perform this key driver analysis to prioritize any potential genes that we may focus on for later studies. So here in the diamond shape means key driver genes, which here I'm showcase the top 2, and I'm coloring the borders of these different genes with red if they are ASD-RV rare variant genes. So here I'm using Safari genes, syndromic, level 0 and level 1 for rare variant of ASD. And I'm using ASD-CV, which are genes linked to loci with a p-value of lower than 10⁻⁻3. So it's kind of a more stringent level than the 5%. I'm labeling the common variants in green. And here you can basically see a lot of red bordered genes across the networks of these 2 different key drivers. And here on this corner, I'm showing you the color patterns of different nodes or genes on this network visualization. The gray ones means that the genes are found to be in this network, but they are not really present in modules associated with the 3 modules presented in these 3 modules. So I'm labeling the 3 modules in different colors to see whether one gene is showing up in one given module across these 3. So basically one thing is that the PCLO key driver is already identified as a major kind of autism rare variant gene according to the Safari database, while the CA-CMB4 is not yet recognized as a ASD or developmental disorder kind of risk gene. But this gene has been indicated in some papers for epilepsy and some sort of neurodevelopmental cases. Cool. So a conclusion for this part is that firstly, I find both 8p significantly correlated modules and the cell type specific DEGs show significant level of enrichment of these neurodevelopmental disorder related common and rare variants. And I also prioritized two key drivers for potential later studies, which are CA-CNB4 and PCRO. And these are shared among the top 8p correlated modules and DEGs. So here I summarize the take-home messages for my results. So firstly, there's a significant changes in cell compositions between the proband and parents at 6 and 9 months of culture. And the DEGs are showing a downregulation of neurodevelopment at 3 months in the proband comparing with the parents. And the top negatively correlated modules from the WGCNA analyses are enriched in neurodevelopment and cell-cell communication related pathways. And at the end, the integrated genomic analyses show a high level of enrichment of both common and rare variants from different neurodevelopmental disorders in our significantly correlated modules and the DEGs we identified in earlier steps. So with this one, I'm also having a little bit of future directions. So if I have an opportunity to kind of still working on more of this data, I think a temporal analysis may bring out more interesting things like how the cells are developing into this in the brain organoid. And also it would be possible to identify potential transcriptional factor driving different lineages or developmental trajectories in the brain organoid. Another thing, wait, I think I accidentally copied this here. Sorry, I have another word. So basically, another thing is to identify how the 8p genotype is affecting tissues across the body. So this can be done by firstly mapping the CNVs related genes based on distance QTLs and also potentially Hi-C data set. And then I have already cell-specific and cell-combined human co-expression modules across 54 different tissues of the human body. So basically, you can also see what are the tissues enriched with the 8p-related genomic signals. So these are the future directions if if I have a future chance. So with this, I'd like to firstly thank the Project AP Foundation, Bina, Whitney, and Ethan for giving me this opportunity to work on this very exciting data set. And I'd like to thank the Moitri lab, Dr. Moitri and Dr. Bassat for the brain organoid culturing and sequencing. And also I'd like to thank my mentor, Francesca, and also my wonderful lab mates for their support across my studies. Yeah, thank you for your attention.
Thank you. We know we're over time, so if anyone has to jump, feel free. But if there's anyone who has any Q&A, just go ahead. And I know we know we're over time here, but if you have any questions.
Thank you, Yanning, for all of your hard work here.
Yeah, thank you.
We appreciate it. And we'll get, I think a lot of us will circle back on trying to regroup on some of the conclusions and future directions of your work. So really appreciate it. And thank you so much, Francesca, as well.
Yeah, you're welcome. This is a rich data set, as any of those single cell data set, 4 time points, lots of cell types. So drawing conclusion is the challenge. But I think Yanning has done a wonderful job to prioritize, trends across in our take-home messages, there are all those, what we think are important take-home messages of reduced neurodevelopment showing at 3 months, and potential, pathway to identify key driver genes. So hopefully you'll find this insightful as well.
Yes, absolutely. And I think that we're also, of course, there's some investigators that join the roundtables generally that are working on trying to expand the N, and then across all the different neuronal subtypes as well. So there's a lot more work ahead of us. I think like projects like this, honestly, just highlight and point out the relevance for that. It's very, very difficult to do these N of ones. Faye?
I love this talk. I think there's so much great stuff there. And I mean, it's so exciting. Right now we're N of 1, but as you say, as we go up and up, like to see which of these trends hold. I mean, and especially, I'm sure you're aware that, there's many different populations within the AP group. So we have some with just deletion, some with inversion duplication, as well as deletion. So it'll be interesting to see how if any of them hold amongst those, or if they're just subtypes, amongst themselves again. But I think my main question from your talk was, at one point, you were sort of showing in one of the slides that Relin was associated with a certain subtype. So I just wanted to make sure that I understood that properly. And I guess my other kind of question around that is, like, I haven't really thought about it this way before, but, do you think it's possible that the sort of the cell subtypes have kind of grown up in different proportions than a neurotypical person would have in our AP heroes, and then so instead of just a cell upregulating a certain protein, that it has more cells of a certain type which produce that protein. And have I understood that correctly? Is that the proposition?
Yes, exactly. So
Okay.
let me answer your questions from a Richard side. So I'm going to answer your second question first, because
Sure.
that's a really important question, and you would like to know whether in AP heroes, whether there is not only up or down regulation of genes in certain cell types, but if there's a total cell type level changes, like compositional changes compared with neurotypical people. So basically that's likely in our suggested from our result, but since like we have a small sample size, so let's say this with caution, but at least if we pull the major cells together, we can see, at least at this 6 months, the two parents look similar, while the proband looks pretty much different from the parent. But it's likely to be like culture issue, but based on the facts from AP heroes, for example, there are corpus callosum, like agenesis, and also there are other more formations in the heart. It's possible to have a different level of cell compositions. So for example, for the corpus callosum kind of agenesis, there may be a decreased number of, for example, oligodendrocytes connecting the two brain hemispheres. But yeah, hopefully, as we have more data, we can safely draw a conclusion of potential cell compositional changes in the AP heroes.
Absolutely. And when you get, sort of hopefully more proband cell types or cell sources to look at, it'll be really interesting to correlate that with MRI data, like real MRI data, right? And reports on these
That's
patients.
possible. Yes,
What's
that's
that?
possible.
Yes,
That's
exactly.
possible to match potential kind of expression with MRI.
Yeah, if we have more traits measured, any type of traits measured from the same patient, we could potentially include them in the trait module correlation analysis and refine also the genetic data integration, and see, to refine which modules and key drivers are more enriched for variants and express, more correlated with different traits.
Mm-hmm. Absolutely, because I know that Madison certainly had an MRI at 2 years old, and yeah, and another one recently. So, it's the sort of data that's available, I imagine.
Yeah, that's rather exciting.
Thank you.
Cool. And for your first question, you are interested to know about the reelin positive cells because in previous like Substack articles, there's previous result talking about there's a increased level of glutamatergic positive and reelin positive neurons in the proband comparing with her parents. And I looked deeper into this cell type because I saw this very same cell type in the proband. And I'd like to know further whether they are potential cohortia cells found really in the neocortex and hippocampus, or whether they are other cell types. So here I put a dot plot showing a lot of kind of gene expressions. So firstly, to say whether where this cell would be expressed if it's in the brain, we would look at the first two markers, which are FOXG2 and also not first two, sorry, which is the FOXG2 and this UNC5C. So the FOXG1 is a marker for the forebrain, for the telencephalon, or the dorsal part of the forebrain. And if the two cells are from neocortex, they would express this gene while they basically express nothing here. While for this UNC5C, which is the cerebellum origin marker, you can see that these two reelin positive cells are expressing very high level of the cerebellar cell marker, and the reelin is here. So basically, if you look at reelin, these two are the top level of expression across all the cell types. And secondly, the CR cells.
And those are the cells that you would not normally see it in, or those are where you would expect to see it?
I would say, since this is a brain organoid, you may see all potential cell types. So yeah, if it's a dorsal cortical brain like model, I would be rather surprised to see cerebellar cells over there, but since it's a brain organoid model, every possible cell types can be there. So yeah, it's normal for a cell type to resemble a cell type from a certain brain area if it's a brain organoid.
If I can add, to this, is that in prenatal brain development, reelin is only expressed by these CR cells. And so in the cortex, and it provides a signal for the newborn neurons to migrate in proper places. And so the cortex has 6 layers, and reelin would generate that signal to say, you go to layer 1, 2, 3, 4, and so forth. So when you think about reelin, you usually think about causal ratio cells, and then they degenerate postnatally, and reelin is still expressed in inhibitory neurons. Our first surprise was that these were not causal ratio cells. So we reproduced the observation that you have more of these cells at 9 months only in the 8p genotype. So there are way more there than in the parents. Although at 9 months, the two parents are quite different, but there is a common shared feature that they don't have a lot of these cells expressing reelin. We first were surprised they were not causal ratio cells, because we were thinking about these organoids as cortical organoids. But then Yanin looked deeper, and we figured out that they actually are expressing cerebellum lineages. So that was first kind of clarification, that there are more reelin expressing cells, but those are resembling granule cells. And so, for example, from the MRI data, I would pay attention potentially if there's any abnormalities in cerebellum, because these data, again, 9 months, a lot of time in dishes, we don't know what happened to this one clonal line in 9 months, but it suggests that in the proband there is an overdevelopment or differentiation towards the cerebellum lineages that you don't observe from the two parents. So perhaps, one could pay attention to any phenotypes that has to do to cerebellum, which beside controlling motor function, and I'm not quite sure if there is a lot of motor deficits in the AP heroes, but
I
it
would say yes.
controls. Okay, so motor function, but also cerebellum is involved in emotional regulation, learning as well. There's a lot more known about the cerebellum now, but mostly is motor control. That's why really lissencephaly, which is a neurodevelopmental disorder where you don't have reeling, one of the major phenotype is also severe motor impairments. So there could be some insight there. Is there a cerebellum phenotype in the AP heroes that maybe could be reflected by this data over proliferation or differentiation of reeling granule cells that suggests that there might be something specific in the cerebellum? So it's something I would pay attention to. That's what I think the data suggests.
Nice, interesting. And then sorry, my final question is like, when you're looking at, the differences in the proband at the sort of 6-9 month point, do you think that, if it would sort of match what maybe the parents would have been at the 4-month point, but we just don't have that snapshot? You see what I mean? So if the proband is just developing slower, you know what I mean?
Actually, yeah, from some part of the ANINS data, it seems like you have a drop in neurogenesis, neurodevelopment at 3 months, so it's slower. But then this data on the granule cells, it suggests there is lower, but also there might be an altered trajectory. So instead of going into this developmental trajectory, they might go to the cerebellar trajectory more, or, in a defective way, because we don't have that in the parents. They don't show up much, those cell type.
Yeah.
Now, whether they would show up later on, we don't know, because I don't think there's data on organoids more than 9 months, or whether it's just a reflection of trajectory deficits. That's why if we were going to do anything more on data analysis from this specific data set, trajectory analysis is something that we think could be insightful, because it seems that some cell types are more specific in the dish for the proband.
Thank you very much. Sorry to hog all that time.
Okay.
Yeah, no
Can I
problem. Thank you
ask
for
something?
wonderful questions.
Can I ask something?
Yeah, for sure.
Yeah, it's great talk, first of all. It's really amazing.
Thank you.
And because someone said trajectory analysis right now. I just wonder how you integrate the data. Did you use the Harmony or such a
I
thing?
didn't use Harmony, but I did try CCA integration by Seurat. And I tried to firstly kind of input and normalize while regressing all the potential batch issues, while I also try to integrate them using CCA, which is canonical correlation analysis provided by
So
Seurat.
how was it like, the merging, like, 9 months samples to the 1 month sample?
Actually, for integration, I'm not sure whether it's the same concept, but for integration, it basically erased cell differences also across time points. So all cells got into a huge lump, and I basically don't think it's possible to get onto that way. So I still kind of just put everything together, regress out the potential batch issue, and then keep on with the clear stuff.
That is really critical right now, right? I mean, you are integrating the trio first, and then you are integrating the different types. And then you're going to do the trajectory analysis, right? So no, I just found it.
Yeah, the trajectory analysis is a really interesting thing. Like, basically, it's one of my interests. But in this scenario, I would say it's rather challenging to do trajectory analysis. So first, because the cell types are rather different, and you basically, in this scenario, you can't, how do I say, for trajectory analysis, you have to try a lot. That's how what I would put it. And in this scenario, also, since there's a significant shift, I'd rather stay with the more traditional DEGs for the first time and leave the potential temporal trajectory analyses later on, because that's basically asking for more effort. And also there may be not real biological issues with the trajectory, because there may be some cell types just dying off, some other cells screwing up,
Like a
and
clonal
it may
solution.
not really be real biology.
Yeah. I really loved your second part.
Yeah, thank you.
Okay, and I thought, it would be really nice, quick, I'll say that if you can compare, those key driver genes in the last figure, CABN, yes, that one. If you can compare, whether those drivers are located on chromosome AP or not, or or whether those drivers are already transcription factors like a GATA4 in this case. That would be amazing. So I think that's gonna make a quick analysis for you, that can get something really intriguing.
Yeah, thank you. So firstly, these two are not transcriptional factors, and I don't really remember which chromosome they are on. But actually, even if they are not really on 8p, they can still kind of regulate 8p or regulate otherwise by kind of their spatial kind of location, because two chromosomes in a nuclei can get really close in location, which can be delineated using a analysis using a kind of technique called Hi-C. And in this scenario, this is a voltage-gated calcium channel, which are usually seen in epilepsy and also other neurodevelopmental disorders. While this PCLO has been characterized in bipolar disorder, major depression disorder, and also I think it's also showing up as the ASD rare variant, but I don't really remember which category it was in, but it was a rare variant according to the Safari database.
All right, well, thank you, Yanning, for your time. And we'll
you.
call it at that. And thank you again, Francesca, for joining, and appreciate everyone's very engaged Q&A for this call. Obviously a testament to how interesting the data set is and what the implications could be. So appreciate everybody's attendance as usual, and this recording will be available on the YouTube channel shortly.
Cool.
Thanks,
Thank you
everybody.
everybody for your attention and your wonderful questions.
Cheers. Thanks, everybody.
Here's-
Bye.